Good morning, students. Today, in plant breeding subject, we have the topic of plant introduction. So today we'll be studying about plant introduction. Actually, plant introduction is one of the, we can say that it is one of the oldest or it is an ancient and the most effective approach to create the genetic variation in plants. Okay, among the plants, because as a plant breeder, we always look towards creating the genetic variation, or we always look towards the genetic variation, how to create the genetic variation. So plant introduction is one of the methods through which we can create the genetic variation among plants. So this was the introduction of plant introduction. Now we'll go into the details. Now, what is the definition of plant introduction? It is, we can say it is a process of introducing the plants from their own environment to the new environment. Now, that plant can be a single genotype or it can be a group of genotypes. Okay, so if we are introducing a particular genotype, or we can say a particular variety or the group of varieties or the genotype of a particular crop species from the environment where it is grown and to a completely new environment where it was not earlier cultivated at any of the times, so that is called as the plant introduction. So I will repeat, it is a process of introducing the plants. It may be a genotype or a group of genotypes from their own environment where they are grown wild, where they're grown by their own, to a completely new environment where they have not been cultivated previously. So the process of introduction may involve new varieties of crop or the wild relatives of the crop species, or it can be a totally completely new crop species for that particular area. Okay, so the process of introduction may involve new varieties of crop or the wild relatives also. It can be a new cultivated variety or it can be a completely wild relative of a particular crop species. So it is grown into a new environment from its own area. Okay, so it is grown into a completely new environment, having all the climatic conditions different. I hope you must have understand. Now again, we will repeat. It consists of taking a genotype or a group of genotypes into a new environment where they have not been grown earlier. Okay, so here you have to listen to this point, that where they have not been grown earlier. So those plants are introduced in those environment or those cultivated area where they have not been grown earlier. And then they are checked for their sustainability, that how they adapt or they show the expression to the completely new environmental area. And it may involve new varieties of the crop. So plant introduction can be a new variety of a crop, or it can be a wild relatives of the crop species, or it can be a completely totally new crop species for that particular area. It can be done within the country. Now this is also very important point, that plant introduction can be done within the country or it can be from other countries of the continents also. Okay, so the plant introduction can be done in the same country, or it can be done in the, that is, plants are or plants parts or the propagules, they're exchanged between the two friendly countries. So as to we always look for the genetic variation to create a huge amount of the genetic variation. So plant introduction is mainly done for creating the genetic variation, and the plant breeders always look towards the plants having a wider genetic, we always look towards a broader genetic variation species. Now the classical examples of the plant introduction in plant breeding were introduction of IRRI, that is International Rice Research Institute rice varieties. So huge amount of varieties have been introduced from IRRI Philippines. Then introduction of sunflower wild species from Russia. So introduction of wild species of sunflower from Russia. So this was also done earlier. And introduction of the oil palm into the Tamil Nadu. So this was also done. So these are the examples of plant introductions. Now, if we go into the history of plant introduction, that how it had started in the field of plant breeding or the agriculture, is that during the 16th century and later, that is the Portuguese and the British and French and Dutch were there in that particular scenario. So in the process of colonization, means the in the process of colonization introduced many plants. So they introduced these Portuguese, Dutch, Britishers, or the East India Company. They have introduced plants from the different regions. Okay, in India, Portuguese introduced new world crops such as maize, groundnut, chili, potato, sweet potato, guava, custard apple, pineapple, cashew nuts, and tobaccos. So East India Company, or the we can say that Portuguese have introduced these new world crop species like maize, groundnut, chili, etc. They have been introduced by Portuguese in India. So they were not grown earlier in India, but many of the varieties have been introduced by Portuguese and the earlier civilizations. Now, a British East India Company introduced. Now, the British East India Company, we see that tea, lychee. So these were introduced by the East India companies to India. Classical examples, if you see in the plant introductions, were introduction of sunflower to USSR from Central Mexico or USA. So the sunflower was introduced from the Central Mexico or the USSR to the USSR, and then the Chinese soybean to North America, and Ethiopian coffee to Central and South America. So these are just the examples where you just have to understand that what was the history of plant introduction and how it had been started. Now, if you think about what is the main objective of plant introduction, okay, so what is the objectives, what are the various objectives of plant introduction? Here we will see that to introduce high yielding varieties, to increase the food production. So the various high yielding varieties which have been grown in some countries, if we want to introduce into our own country, and in order to, we are doing this thing to enhance our yield. Okay, yield production, we are trying to increase the yield, because in order to feed the huge amount of population, we always have to work upon increasing the yield production. Okay, so high yielding varieties, they have been introduced from other countries to our countries, like in example in case of rice and wheat, to enrich the germplasm collection. So this is also very important, that we do plant introduction to enrich our germplasm collection. Germplasm collection is nothing but the collection, the huge gene bank of various plant species having there. We should have, means to create the genetic variation. Okay, so there are many institutes which have the germplasm collections of almost all the crop species. So to broaden our genetic variation, we we do plant introduction to enrich the germplasm collection, like we have done in case of sorghum and groundnut. Then the third point is to get new sources of resistance against biotic and abiotic stresses. If we are looking for a particular resistance genes for maybe for insect, pest, or for various biotic factors, so we can go for plant introduction. Okay, then to introduce a new plant species by creating ways to build new industries. So by the introduction of oil palm in Tamil Nadu, so we have also promoted the oil industries also. So this is also the objective of plant introduction. Type of plant. Now if we look into the types of plant introduction, so there are two types of plant introductions broadly: primary introduction and secondary introduction. And based on the utilization, again the introductions can be classified into two types, that is direct introduction and indirect introduction. But broadly there are two types of introduction, that is primary introduction and secondary introduction. Now we will go into the details of it. Primary introduction. Now what is primary introduction, and what are the various examples of primary introduction, which are very important in terms of understanding, is when the introduced variety is well suited to a new environment. So if any particular variety is introduced into a completely new environment, okay, and if it is well suited, if that particular new environment is well suited for that particular variety, and we don't have to do any alterations of the genotypes, means any genetic manipulation or any change in the through hybridization or selection, we don't have to do anything, but that particular variety, that environment, that new environment, it well suits that particular introduced variety, then that is called as primary introduction. So when an introduced variety is well suited to a new environment, then it is called as primary introduction. Then it is released, because because we don't have to do, as a breeder, we don't have to do any changes in that particular variety, because it is well suiting to the new environment. So it is released for the commercial cultivation without any alteration of genotype. So doing any change, we don't have to do any change in the alteration of the genotypes, and we can release it for the commercial cultivation. So the examples of these primary introductions are dwarf wheat varieties of Sonara 64. That means wheat varieties, the dwarf varieties of wheat like Sonara 64 and Lerma Rojo. So these were the classical examples of primary introduction. So these are asked in various of the competitive exams. So you have to remember that the primary introduction examples are Lerma Rojo and Sonara 64 or dwarf wheat varieties. Then dwarf rice like Taichung Native 1, TN1 and IR8. So these were also the examples of primary introduction. Various IR series from IRRI Philippines have been the examples of primary introductions like IR8, IR20, IR34, IR50. So these are the rice varieties and the oil palm varieties. They were introduced from Malaysia, and Mahsuri rice from Malaysia. So these were all the examples of primary introductions. Now going into the second part, that is secondary introduction. Secondary introduction, in secondary introduction, what happens is all those introduced varieties which are subjected, all those introduced varieties which means there are certain changes in the expression of the characters when they have been grown in the new environment. So the expression, their characters is not that much good that we expect. So, the plant breeders, what they do is to create certain amount of genetic variation, or we do selection, and then we also go for hybridization. That means crossing with other type of genotypes and transferring some of the desirable traits. Okay, either we can transfer those desirable traits from the local cultivars or the local varieties. So the introduced variety is subjected to selection, or it is used in hybridization, that is crossing program with the local varieties. So why we are doing here hybridization or crossing? Because they are not performing as per expected. So we want to transfer certain desirable traits or the genes for either for the insect resistance or pest resistance or other type of stresses like biotic and abiotic stresses. So we go for selection, and then we go for hybridization or the crossing program with the local varieties. Okay, so that is called as secondary introduction, where we are doing certain type of gene manipulations, we are doing certain type of crossing and hybridization. Okay, so they are not, means as such, they are not released for commercial cultivation. Certain amount of crossing and selection is done first. So those varieties are called as secondary introduction varieties. So to get the improved varieties with some new characters, we do this type of secondary introduction. We go for selection and then we go for hybridization. Now the examples of secondary introduction are like Kalyan Suna and Sonalika varieties of wheat. Earlier they are the examples of secondary introduction. They were introduced from Simvaiti, Mexico. Now, what is the procedure for plant introduction? Ways of procurement of plants or germplasm. Now, what is the procedure? How we go for plant introduction? That you should also know. So, there are certain national and international organizations which are involved in the plant introduction, and there are certain rules and regulations, and the phytosanitary certificates have to be issued, and there are certain quarantine rules also where all the breeders, they have to follow before introduction of a particular plant part from other countries. Okay, so ways of procurement, how to procure a plant or a germ plasm, what is the procedure? So requisition for the introduction of new plant, crop plant, or the new varieties, they should be submitted to NBPGR. Now here NBPGR, that is a National Bureau of Plant Genetic Resources. So this is the national organization where all the introduced crop plants or the species, they are submitted there only in the NBPGR organization within the country. So this NBPGR is an institute which is in our country, which is a national institute, and if we go into the international institutes, then IBPGR, that is the International Bureau of Plant Genetic Resources. So this is the international organization that is located in Rome, Italy. So this is responsible for all type of plant introduction all the rules and regulations we have to follow there. So all those plants and propagules or those new crop varieties or the germ plots, they have to be submitted there first. The material after the submission there, the material may be obtained on exchange basis from the friendly countries. Now, whatever the material, or we can say whatever the genotype or a group of genotypes that are introduced from other countries, either we receive it from based on the friendly basis from the other countries or on the exchange basis. Certain genotypes or the genes or the varieties, they are exchanged also, or the other way is through the friendly basis, friendly countries, or the materials can also be purchased or obtained as free gift from individuals or organizations. So certain plant breeders, they introduce, means they give the other breeders for a particular evaluation or the multilocational trials for to the other plant breeders. So based on the friendly environment, or from they are exchanged from one organization to the other organizations. Now second is packaging and dispatch. So after the plant material have been procured, then it is packaged and dispatched. How it is done? Part of the plant. So what is actually, before that you have to understand that what is a propagule. Now propagule, it can be a seed, or it can be a tuber, potato tuber, or anything. It has to be a certain plant part. Okay, part of the plant which is used for the propagation of that species is called as propagule. So it is properly packed, and then it is dispatched. Then propagules. What are propagules? Propagules can be a seed, tubers, runners, suckers, solons, bulbs, root cuttings or buds, etc. It can be anything. Any part of the plant can be called as propagule, which is used for the exchange purpose or for the plant introduction purpose. And the propagule are packed carefully, and then it is dispatched to a particular organization where it has been required. Now the entry and plant quarantine. Now before the dispatch, that particular plant propagule is entered, and everything it has given, that organization gives a particular number to that, and before releasing it has to pass the various phytosanitary certificates also. So on receipt of the material, once the material is being received, the entry inspection is done by the country. So whichever country receives that particular plant product, it does the entry part of for a particular contaminant. So there can be certain amount of contaminants present or admixtures present in that particular seeds or the plant part, or if that particular plant part or the propagule can be infected by the insect diseases or nematodes. So it is properly checked, and the materials, they are also treated by insecticides, pesticides, fungicides, nematicides and other used. So before it is released for the trial purposes, all these things have to be properly inspected. And then last point is cataloging. Introduced specimen. So whatever the specimen which has been introduced from other country has to be, means a particular number is given by to it regarding the species, then a variety, then it is written that which variety is there and what is the place of origin, and the data, all the data is being recorded like this. So cataloging, properly cataloging is also done. So all these regulations, rules and regulations have to be followed before it is released for the commercial cultivation. And the final step is evaluation. Performance at the different sublocations are evaluated. So then that particular plant material is being given to all the to different organizations for their multi-locational trials, and then it is evaluated at the different environments, because the different locations will have the different environmental conditions. So it is evaluated at different environments also. And then final step is multiplication and distribution. After all these procedures, then it is released for multiplication, and that particular genotype is released for multiplication, and because it has to be produced in the large amount, and then that is distributed to the particular users. Okay, so the promising introduced materials are propagated and then released as varieties after necessary trials. So different types of multi-locational trials are also done at the different locations before releasing it into the different varieties. Now we will look into the agencies of plant introduction in India. So what are the different agencies which are responsible for plant introduction in India and at the international level also? So the IPGRI, that is the International Plant Genetic Resources Institute, which is located in Rome, Italy. It deals with the introduction of plant material on the global level. So on plant introduction at the global level is done by an international organization, that is IPGRI, that is located in Rome, Italy. Now in India, we see in the year 1946, IRI New Delhi has introduced NBPGR, that is a National Bureau of Plant Genetic Resources. It is one of the big organizations that is responsible for the various types of plant introduction, and this organization, that is NBPGR, was started in the year 1976. So these years and the full forms you should remember. In India, there are two more organizations apart from NBPGR at the IRI. So there are two other organizations, that is FRI, that is a Forest Research Institute, that is mainly responsible for introduction of mainly the forest plant species, and the other one is Botanical Survey of India, that is the BSI, that is located in Calcutta, and FRI in Dehradun. So these are two more organizations in India which are responsible for various forest, medicinal and aromatic plant species for their plant introduction. FRI deals with the introduction of forest trees, I have told you, whereas Botanical Survey of India is concerned with the introduction of medicinal and botanical plants. So FRI mainly deals with the forest species, whereas this Calcutta, that is a Botanical Survey of India, that is in Kolkata, is mainly responsible for introduction of the medicinal and the botanical plants, like the medicinal plants and the various ornamental plants which are responsible for their aesthetic values. Okay, so various crop research institutes also introduce plant materials, but only through the NBPGR New Delhi. So various research institutes, they can introduce the plant materials, but only they have to follow all those NBPGR rules. It has to be submitted into the NBPGR first, and thus NBPGR coordinates the work of plant introduction in India. We can say that NBPGR is the prime organization which is responsible for all the work which is related to the plant introduction in India. Now, what I have already told you, that all the rules and regulations have to be followed, like quarantine, or if a particular plant propagule is introduced from other countries, so it has to be quarantined. That means it has to be kept for about a minimum amount of 15 days, and it has to pass the phytosanitary certificate before it is released for the multiplication and distribution among the scientists or the organizations for their multilocational trials. Like, it is being checked for any type of admixtures present, or any type of insecticides, nematodes. So because earlier there are certain cases where where the seeds were mixed with the weed seeds, and like the classical example is about the lantana camera. So these were the weeds which have prevalent in India now, because they have been bought. These were not earlier grown in India, but they have been introduced as a form of admixture from the other countries when the seeds were being imported. So all those quarantine rules have to be followed. That is for and there is a Destructive Insect and Pest Act, that is DIP Act of 1914. So it has to be followed. Now we will look into the now we have understood that what is plant introduction, what is the procedure. Now we will look into the merits and demerits, like what are the advantages of plant introduction and what are the disadvantages of plant introduction. Now, if we go into the advantages of plant introduction, is that first very important point is it provides entirely new crop plant to a place. So it through plant introduction, we get entirely a new crop plant to a particular place, because it has been introduced from other area, and it is well suited. If it is well suited to that particular area, so we get a completely new crop plant to that particular place, and indirectly we can also increase our yield through this. And superior varieties may be originated directly or after selection or hybridization. And suppose we go for selection and then hybridization or crossing with some of the local land races, and certain desirable characters, they are being transferred into the introduced variety, then indirectly what we are getting is a superior variety. Okay, so superior varieties may be originated also, and The germplasm, we are also broadening our germplasm resource. Okay, we are widening our germplasm resource, the genetic variation we are creating. So germplasm collection, maintenance and protection of the genetic variability is also one of the important advantage of plant introduction. And the point is, it is the most quick and economical method of crop improvement, because all those hybridization, selection, crossing, and various breeding methods, they are time consuming. It takes numerous years for getting a particular variety, to develop a variety and to release a variety for the commercial cultivation. So it is one of the quick and the economical method of crop improvement. We can do the crop improvement by simply plant introduction of a particular variety from one country to the other country. So it is one of the quick and the economical method of crop introduction, when introduced material can be used directly, if it is a primary introduction we are talking about. Then introduction of some of the varieties to the newer areas may protect them from some of the diseases. So if we are introducing a particular variety to our area where it is not affected by a particular disease, so indirectly we are protecting that particular variety from attack of from the various biotic and abiotic factors also. Now, what are the disadvantages? So there are certain disadvantages also related to plant introduction. We will look into it. Now, the most important and the most prominent disadvantage of plant introduction is that while introducing a particular genotype from other countries, we with the admixtures, in the admixture form with the seeds, we all sometimes the seeds are also mixed with the seeds of weeds also. So this was seen in case of weeds like Argemone mexicana, Eucarnia, Lantana camera. So they have been introduced from other countries through the seeds only. So these are very prominent weeds nowadays in India that spread widely in most of our ponds. So these have been introduced through seeds, through plant introduction from the other countries. And the fungal diseases like late blight of potato, flag smut of wheat, coffee rust, punchy top of banana. So these were all the fungal diseases. They have also been introduced through plant introduction only. Okay, they have been introduced in India along with the plant materials. Okay, when the plant materials have been introduced from other countries to our country, so these were the fungal diseases, they have been introduced along with that plant materials. So all those phytosanitary certificate has to be cleared nowadays through NBPGR, and it has to follow all the rules and regulations of DIP Act and all the rules of NBPGR before it is introduced into for multilocational trials, and it is finally released to the users. Many insect pests like potato tuber moth, woolly aphis of apple were introduced in India. So likewise, through plant introduction only, potato tuber moth, woolly aphis of apple, these were also introduced in India through this. Okay, so here I will end today's topic. I think most of us we have covered one. Okay, here now in last we will see about the achievements in plant introduction. So major achievements, like most of the we have got large number of varieties like potato, maize, groundnut, chilies, coffee, rubber, guava, grape, various other these crop species, they have been grown nowadays through plant introduction only. And new crop species, new crop varieties like direct multiplication, they have been released as new varieties. So various varieties of rice like Sonara 64, Lerma Rojo, and dwarf wheat varieties like TN1 and TN1, IR8. So these were the wheat and rice varieties. They have been introduced in India through direct introduction. Okay. And desirable, like in case of wheat, Kalyan Suna and Sonalika, these were also the desirable varieties. They have been introduced in India through introduction only, through plant introduction only. So these were the major achievements that we have got through plant introduction. So here I will complete my today's topic. Thank you. See you in the next video. Thank you.